Hello everyone. I'm Mohammad Tariq Sayed, Assistant Professor, College of Agriculture, Parul University. Today we are going to start a very interesting and informative and productive webinar on Sanger sequencing, past success and current applications. So DNA sequencing, it includes any method or technology that is used to determine order of, we can say that 4 bases of the DNA: adenine, guanine, cytosine, and thymine. The advent of these rapid DNA sequencing methods has greatly accelerated biological and medical research discoveries. The Sanger method of DNA sequencing, it is developed by the Frederick Sanger and his colleagues in 1977. And currently it is most widely used sequencing method for approximately last 14 years. So DNA sequencing technologies, it will be a game changer in the field of biology as well as in the field of medicine also. So without wasting time, let's introduce and welcome our chairman of today's webinar, former Dean in Principal, currently HOD of Department of Horticulture, Dr. R.J. Jadhav, sir. Thank you, sir, for your kind support. We have Dr. Pooja with us. She has done her graduation and a post-graduation in the field of biochemistry and biotechnology, and completed her PhD from the Anand Agriculture University. She has 5 years of research experience at Animal Biotechnology Department, Veterinary Science College, Anand Agriculture University. She has 10 plus research publications in the field of biochemistry and the biotechnology. She won Young Scientist Award, and we are lucky that Dr. Pooja has given time to us. Thank you, ma'am, for us. Without wasting time, I would like to invite Dr. Pooja, ma'am. Please share your valuable words to our audience. Ma'am, the platform is yours. Thank you.
Good afternoon to Dr. R.J. Jadhav sir, all the faculty members and dear friends. I am myself Pooja Magad. I am here to present my webinar topic on Sanger sequencing, past success and current application. We'll start with the introduction. DNA sequencing is the process of determining the sequence of nucleotides in a piece of DNA. On the earth, all the living organisms having the genome, and this genome is made up of nucleotides only. That is the ATGC, adenine, guanine, thymine, cytosine. But the arrangement or the sequence of this nucleotide that makes them different, like human being, plant, and microorganisms. So study of this nucleotide arrangement is DNA sequencing. Nucleotide order determines the amino acid order and by extension protein structure and function. So for proteomic study, we need to first know the DNA sequence that has been carried out with the DNA sequencing. DNA sequencing technique are the key tools in many fields. The fields which are related with the proteomics, transcriptome study, and the genomics that need to know the the nucleotide sequence of that particular gene they are studying. So different science are receiving the benefits from these techniques. The application of this sequencing techniques of DNA sequencing is in the field of archaeology, molecular biology, evolutionary biology, metagenomics, epigenomics, forensic identification, DNA barcoding, and genetic testing. All this field required the sequencing study. First, why DNA sequencing is important? As I said, on the earth, all organisms having the genome, and that are made up of different genes, different repetitive sequences, the exon, introns, that all having the functions. And how we can know the function of that this DNA fragments? By confirming their actions. So by DNA sequencing technique, we know the sequence of that nucleotide DNA fragments, and to know its confirmed action, make prediction of its function. So DNA, whatever the gene we study, we need to predict this. We need to confirm its action, and by prediction, by knowing its function, to facilitate the manipulation of DNA molecules. If we study some gene or the DNA fragment, we need to and we give some treatment to that plant or an drug treatment to animals, so whether there will be any change of at the genetic level. So we have made manipulation to that DNA molecules, and we need to know whether at the genetic level the manipulation is done or not. That can be confirmed by the DNA sequencing only. To compare the two molecules like normal and mutant, in nature there will be of in a simple same species or two different individuals, there will be of different characters, like having a normal character and mutant character, but initially they are coded by the same gene, but that different can be studied by using the DNA sequence, because of that single nucleotide change will cause it mutant and normal one is a wild one. And also the detection of the exact order of nucleotides within any fragment of DNA. By using all this, we can create the whole map of the genome. Now coming to the history, prior to the 1970s, there was no any satisfactory method of sequencing DNA. So scientists has to depend on the reverse genetics to finding out the sequence of DNA. Reverse genetics, it means nature having previously, when there is no DNA sequencing, the scientist will analyze the observe the phenotypic character, and based on that different phenotypic character, that goes to back to the study and come to the genome. Here also the problem is there degeneracy of codon, because phenotypic character, that is because of protein, and this protein is coded by the 20 amino acids, and this 20 amino acids back to study, it is coded by 64 codons, and these 64 codons again are coded by the 4 nucleotides only. So there will be again degeneracy of codon, means 20 amino acids are coded by 64 codons, so there will be definitely overlapping of this codon, and thus how this protein study, this reverse genetics will create some error by or we can say a difficult task for scientists to study at the genetic level. By discovery of double helical structure of DNA by the Watson and Crick in the 1953, and in 1977, Maxam and Gilbert have given the chemical degradation method, and Sanger given the dideoxy sequencing method. And this 1985 Kelly Mullis, this PCR in vitro replication DNA, all three discovery that made possible of automated DNA sequencing technology. So in 1986, Hood and Smith announced a first automated DNA sequencing machine. With the help of that, DNA sequencing has been carried out. Now the generations of sequencing. There are 3 levels of generations DNA sequencing technology. First generation include Maxam and Gilbert and Sanger dideoxy chain termination. This is the chemical degradation method and dideoxy chain termination method. Second generation include the Roche genome sequencer, also called the pyrosequencing, Applied Biosystem Solid 3 Plus system, and third one is Illumina genome analysis system, Helicos biosystem, and Pacific biosystem. This all system are coming into the next generation sequencing. With the help of that, whole genome can be carried out. Whole genome sequencing can be carried out. And third generation include the ion torrent nanopore sequencing and likewise other techniques. First, next we are focusing on the DNA sequencing method of first generation sequencing, that include the chemical degradation method given by Maxam and Gilbert, and second one is chain termination, also called the dideoxy Sanger sequencing method. Because of so many limitations to the chemical degradation method, in that Maxam and Gilbert have used the hazardous chemicals, and also the DNA fragment length is of the around 300 base pair. That is not enough to predict the function or the sequence of any gene that is of around 1000 kb, around thousands or hundreds of kb. So that limitation is overcome by chain termination Sanger dideoxy Sanger method. In 1977, Maxam and Gilbert published a paper called A New Method of Sequencing DNA. And second, and the same year, Sanger also published a paper called the DNA sequencing with the chain terminating inhibitors. Now coming to Sanger sequencing, it is also called the dideoxy sequencing or the chain termination method, because it is the same like the PCR only, in vitro amplification of DNA fragment. But here a unique nucleotide characteristic is there, addition of dideoxy nucleotides with respect adenine, guanine, thymine, cytosine, all 4 nucleotides with the dideoxy. Means at the 3' OH, there will be reduction of OH. So further it is not further amplify, not allow to extend the nucleotide, and chain will terminate it immediately. Then further use a DNA polymerase to synthesize a complementary copy of a single stranded DNA. region of the DNA up to about the 900 base pair in the length can be sequenced using a method called the Sanger sequencing or the chain termination method. In 1975, it is called the plus and minus sequencing of 5386 base pair phage that utilize the E. coli DNA1 and DNA polymerase from the bacteriophage T4 with the different limiting triphosphate. Sanger sequencing was developed by the British biochemist Fred Sanger and his colleague in 1977. In Sanger sequencing, the target DNA is copied many times, making the fragments of the different length. As I told in the previous, here in the Sanger sequencing, in the one step, it is also called the cycle sequencing. It means in a PCR reaction kind of technique, the input DNA is amplified 1,000 times, and it is also added with the dideoxynucleotide. So it is get terminated at the respective nucleotide, that DDATPs like ATGC, and produces thousands of copy and called it as a collection of sequencing ladders. And for the fluorescent chain terminators, nucleotides mark the end of the fragment and allow the sequence to be determined. As in the reaction mixture, dideoxynucleotide is there, and it is also labeled with the fluorescent dye or the radio labeled dye. So along with the primer annealing and amplification with the normal nucleotide and termination with using the dideoxy, there will be of thousands of copy for the dideoxyadenine, dideoxyguanine, dideoxycytosine, dideoxythiamine. And these terminated nucleotide also are labeled with the fluorescent dye that further can be detected by the detector system, and that will be given in the further in the chromatogram format. The Sanger sequencing is in a wide use for the sequencing of individual pieces of DNA, such as fragments used in the DNA cloning or generated through the polymerase chain reaction. Sanger sequencing are using in a many DNA fragments of around 900 base pair to short stretch of the DNA can be sequenced, and also it is used in the cloning system. Means in a cloning there will be insert DNA of 1 kb to 20 kb to 40 kb. This DNA fragment can be sequenced by further chopping this DNA and further making a fragment of 1 kb, and that can be further DNA sequenced. Now the ingredients for the Sanger sequences are similar to those needed for the DNA replication of an organism or for the polymerase chain reaction, which copies the DNA in vitro. They include a DNA polymerase enzyme, as like in the PCR. In the Sanger sequencing, there will be DNA polymerase that amplify the primer further complementary to the template DNA. A primer, which is a short piece of the single-stranded DNA that binds to a template DNA and act as a starter for the polymerase. In the normal in vitro, that PCR amplification, we generally use forward and reverse primer that define the particular size of that particular gene. But in case of the Sanger sequencing, that already having a limitation of 901 kb base pair DNA fragment. So only one primer, whether it is a forward or reverse, one primer is used, because DNA is itself a double-stranded DNA. So we in that case, we can consider it as a 2 DNA fragment because they are complementary to each other. So at a time, only one forward or reverse primer is used for the one sample. The 4 nucleotides, that is DATPs, DTTP, DCTP, and DGTP are used, and the template DNA to be sequenced are used. However, a Sanger sequencing reaction also contains a unique ingredients called dideoxy or the chain terminators. The version of all 4 nucleotides, means normal DNTPs are there in the reaction mixture, but these unique ingredients are the dideoxynucleotides, dideoxy-ATP, dideoxy-TTP, dideoxy-CTP, and dideoxy-GTP, each labeled with the different color of dye. Although it is in the 4 different tubes, they are labeled with the different colors. So further analysis and further reading of this complementary sequenced DNA can be easy by identifying the different color. Special requirement for the sequencing reaction is the DDNTPs. that possess the 3' H hydrogen group instead of 3' OH compete in the reaction with the normal DNTPs and produce no phosphodiester bond. In a normal reaction mixture, this DNTPs is used. It is having a deoxyribose sugar at 5' a phosphate group at first prime, there is a base attached, but at the third position there will be of oxygen hydroxyl group. On that further polymerization is happen, DNA polymerase get amplify, extended this DNA fragment by adding the further nucleotide over here. But in case of the Sanger sequencing, the dideoxynucleotide, there will be of dideoxyribose is here. Why? Because at the second position OH is not there, and also at the third position no OH is there, only H is there, so it is called the dideoxynucleotide. So there is no any way to polymerize further this dideoxynucleotide further any normal DNTPs or dideoxynucleotide. Here double-stranded DNA depicted in a form like 5' to 3' one DNA fragment, and the complementary fragment in the opposite side from 5' to 3'. Here also a nucleotide that having a third at 3', instead of OH there will be of only hydroxyl group. So that absence of OH group, that will not allow further reaction to get a further nucleotide to add, and further the reaction is terminated. Whenever the radiolabeled DNTPs are incorporated in the chain, the DNA synthesis terminated because of that 3' H group, not the OH group. Each of the 4 reaction mixture produces a population of DNA molecules with the DNA chains terminating at all possible position. While preparing this reaction mixture, initial components are the DNA polymerase, DNA to be synthesized, DNA to be sequenced, then DNTPs. All that components are there, but for one reaction there will be preparation of 4 tubes, and respective dideoxynucleotides are added in a 4 tubes. Like in a one reaction mixture there will be of DD-ATPs, then second tube having the DD-GTPs, then third tube having the DD-CTPs, and the fourth tube having the DD-CTPs, and then allow the reaction mixture to proceed. Extension product in the each of the 4 reaction mixture and with the different radiolabeled DNTPs depending on the base. While allowing the reaction, there will be of how reaction will done. DNA to be sequenced are allowed to anneal with the primer, and there will be amplification by using the normal DNTPs, and then further terminated by the respective DDNTPs, which is added into the respective tube. So next, each reaction mixture is electrophoresed in a separate lane, means for one sample there will be of 4 lanes at a high voltage on a polyacrylamide gel. The patterns of the bands in the each of the 4 lanes is visualized on the X-ray film, as it is these 4 deoxynucleotides are labeled with the radioactive that can be further detected, visualized using the X-ray film. Location of bands in the each of the 4 lanes indicate the size of the fragment terminating with the respective radioactive labeled DNTPs. When we are allowing to electrophoresis of this 4 reaction mixture in the sequencing gel, these are electrophoresed, the molecules get separated based on the charge to mass ratio. Here the all molecules having the same charge, but they are different with the one nucleotide only. Why? Because the DNA, the primer and the amplification with the one nucleotide terminated. Then second band having the primer amplification with the second nucleotide termination. Likewise, all the thousands of sequencing fragments, they are terminated with the irrespective of this DNTPs, 4 different DNTPs, but they are one nucleotide difference are there, and that one copies are present in a hundreds of copy in that reaction mixture. Then DNA sequence is deduced from the pattern of the bands in the 4 lanes. Here all the This Sanger sequencing, cycle sequencing reaction, the detection system are depicted in the picture. Here, the single-stranded DNA are, to be sequenced are taken and allow a primer to anneal to a particular position. Then DNA polymerase further polymerize this primer by using the DNTPs. The all DNTPs are there in the all 4 tubes. So further normal further polymerization is happen. Then all these reaction mixture are separated in the 4 different tubes. 4 reaction mixture are there, and they are again specialized with the unique DNTPs, like one tube having the DATPs, second have a DDGTP, then DDCTP, and DDTTP. Means if when we are allowing this reaction mixture to amplify, this will amplify like primer amplification, and the further if it is if it is A, then terminated with the DDATPs. Then polymerization, then second, if A is there, adenine is there, it is terminated with the DDA adenine. Likewise, this reaction happen with the all respective nucleotides. Then these all 4 nucleotide reaction mixtures are loaded into the sequencing gel in the 4 different lengths, then allowed to electrophoresis. And the reaction mixtures are electrophoresis based on the charge to mass ratio. All molecules having the same charge, but they are different with the one nucleotide. So the shortest DNA fragment that having the primer plus amplification with a terminated with the one nucleotide, the first nucleotide that having the lowest molecular weight, so get run faster. Then second one is the primer plus second nucleotide that get terminated, and that is bigger than one nucleotide only. Likewise, electrophoresis is happen for all the DNA amplified product in a sequencing gel, and reading has been carried out from bottom to the top. So with the all 4 tubes and the amplified product and the sequencing and the electrophoresis of this sequencing gel, reading can be reading done with from bottom to the top, and this complement and this sequence is complementary to the original template which we need to sequence. So thus how we can have the sequencing that DNA segment. Now here also the merits and demerits, means advantages and disadvantages of this radio labeled dideoxy Sanger sequencing. Means the benefits are the reactions are generally faster, does not require any hazardous chemicals like in the previous one, like in the chemical degradation method, and can be used with the limited quantity of the DNA. Longer read length and low cost and high accuracy is done with this DNA sequencing technique, and automation is also possible. But there is also some limitations are there, like longer time, because this radioactive labeled amplified product are run in the sequencing gel. So further electrophoresis and further analysis and further depict the sequencing nucleotide sequence, that will be time consuming, so it will take longer time. Also the size of DNA fragment and radioactive molecules are used over here, and throughput was low, typically of around 300 base per run we get. To overcoming this problem, automated dye-terminator DNA sequence has been done. Means some modification is done in this Sanger sequencing. Here, automated DNA sequencing employs the same general procedure, but uses the DDNTPs which are labeled with the fluorescent dye. Previously, the DDNTPs are labeled with the radioactive, and they are performed in a 4 different tubes for a one reaction, for the one sample. Here in this case, DNTPs are labeled with the fluorescent dye, and that all fluorescent dye are labeled with the 4 different dyes. This combined 4 dyes in a one reaction tube and electrophoresis in one length on a polyacrylamide gel or the capillary containing the polyacrylamide gel. Means in a one reaction mixture, all 4 dye containing DNTPs, means DNTPs are labeled with the 4 nucleotides are labeled with the 4 different dyes, and that all together are put in the one single tube, one single reaction mixture that having the one sample, and then that can be further cycle sequencing is allowed, and then further electrophoresis is carried out into the capillary electrophoresis. Then further detection is carried out in the UV laser detects the dyes and reads the sequences. Here the detection system for the fluorescent dye is the UV laser. Sequence data is displayed as a colored peaks like in a chromatogram that corresponds to the position of each nucleotide in the sequence. Here these 4 dyes are detected by the UV laser, and they are presented in the system like a 4 different color peak, and they are presented in the chromatogram. So that is also again a somewhat easy task to analyze the data. Throughput is high up to the 1200 base pair reaction, like in the previous one it is of 300 base pair only, and 96 reaction in a one plate every 3 hours will with the capillary sequencer. This says that here automated system is also there. Fluorescent dye we are using in a one reaction, and also in a one plate 96 reaction means 96 samples has been carried out at a once, and that is the DNA fragment of having the 1200 base pair DNA fragment, and that all procedure has been carried out with the 3 hours only with the capillary sequencer. Most automated DNA sequencer can load robotically and operate around the clock for a weeks with a minimum labor. It also says that automated system means we need to we have to prepare only the plate of 96 reaction, means 96 samples, and we have to keep that plate in a genetic analyzer. The system is called the genetic analyzer. In that we put that plate in that system, and robotically that system is taken this tube, and this all is programmed by the nearby computer having that software that govern all this robotic program. The instrument automatically take this plate, that instrument automatically will cleanse these capillaries with the use of water and buffer, and automatically it will take the samples. Spectral properties of some fluorophores used in the DNA sequencing. A set of the 4 different fluorescent dyes that allow the all 4 reactions to be separated in a single lane. As I told, in this system, the all 4 fluorescent dye are used in a single reaction mixture, so that all reaction will happen in a single sample and analyzed with this in a single lane, means single tube only. The 4 acceptor commonly used in a dye primer chemistry are the FAM, Joy, Tamra, and ROX. These are the 4 dye which are labeled to the respective nucleotides. FAM giving the green color, Joy giving the red color, Tamra giving the black color, and ROX giving the yellow color. The major advantage of energy transfer dyes are that they can be almost evenly excited by a single wavelength. As I told, in the fluorescently dye, there will be at the end detector system uses the laser light of a single wavelength, that is of 488 nanometer. That single wavelength laser light fluorescent or emit and receive the light of emit this all 4 dyes at the same time, and detect its emitted light and converted it into the color peaks and give us in the chromatogram format. These dye also show the uniform electrophoretic mobility and high fluorescence intensity. So these are the advantage over the radioactive molecules. Now, how this fluorescently dye labeled system is done. So in the PCR reaction, all these are the single stranded DNA to be sequenced, and primer is annulled, and further one nucleotide is get terminated in a one single tube that having the all 4 dideoxy nucleotides. So further, if in complementary to the adenine, there will be a dideoxy nucleotide thiamine is there. Then likewise, this collection of DNA fragments like sequencing fragments are there in a single tube, and then further these reaction mixtures are allowed to electrophoresis in a capillary tube. In case of sequencing gel, that can be dissolved with the sequencing gel that is made with the 7 molar urea and 4 to 8% acrylamide gel is prepared, and 200 volt constant temperature of 65 is given. In case of the capillary electrophoresis, this allows the use of the high electric field, but although small capillaries dissipate the heat that produced within it. Large electric field allows for an efficient separation and reduction of the separation time. Capillaries, capillaries having the acrylamide gel polymer gel that allow this reaction, the sequencing product fragments that allow to separate it based on charge to mass ratio of these fragments are differentiated because with the one nucleotide, and that efficiently separated this sequencing product. Now thus this whole system is look like this. The instrument is known as, we call genetic analyzer, Applied Biosystems 3500 genetic analyzer, attached computer, and that having a software. By this software, this whole system is operated. We need to keep only a 96 well plate within it, and rest of the function is robotically handled by the software which is there in the computer. Then capillaries are automatically washed with the water and buffer, and samples are taken in the capillaries by operated by the system. And the capillaries are within this capillary, the acrylamide gel is there, and that provides the matrix for separation of this sequencing fragment. And here it is of detector that detect whatever the sequencing product resolved based on charge to mass ratio, and further that fluorescent dye emit the light according to its fluorescent dye attached, and it will detect it in a form of the colored peaks in a blue, green, red, black likewise, and give us in the form of the chromatogram. The chromatogram is look like this. Like thymine gives the red color, guanine gives the black color, adenine gives the green color, and cytosine gives the blue color. Now the genome sequencing projects are short history timeline, means past successes with the Sanger sequencing. In 1974, Robert Hawley determined the complete sequence of RNA of a bacteriophage called the MS2. 1977, Frederick Sanger in England determined the entire sequence of the bacteriophage 174. In 1784, the entire sequence of HIV virus genome was determined by Kyron Kopp. In 1995, the first genome of free living organism, that is the Haemophilus influenza, was completely sequenced. In 1996, the complete genome sequence of E. coli bacteria was sequenced. In 1999, the complete genome of fruit fly, that is Drosophila melanogaster, was sequenced. In 2000, the first plant genome, Arabidopsis thaliana, was published. In 2001, the first draft genome of entire human genome was published. In 2002, the genome of rice, that is the Oryza sativa, was sequenced. And in finally 2003, the finished human genome was published. Although this all genome projects has been carried out with the next generation sequencing also, but the main important, the gap filling, the important gene, the repetitive sequences, the important areas of genome, that all are again sequenced by Sanger sequencing and confirm this genome map, and then they are published. Now the Sanger sequencing current application. Sanger is considered as the gold standard sequencing method for validating the sequence of specific genes, including those already sequenced through the NGS. Means from previously Sanger, from 1977, Sanger are used up till now. Why? Because Sanger is giving a quality of nucleotide sequences of QC value of 20 or 20 to 25. It means in a DNA sequencing technology, the QC value of particular nucleotide, that defines the quality of DNA sequencing. So NGS, although NGS giving millions of copy of a whole genome very nicely, but although there is a small gaps in the important regions, that repetitive sequences that are not nicely sequenced by using the NGS technique. The main Importantly, NGS are not amplifying the repetitive sequence very accurately. Means if repetitive sequences are there, NGS accurately cannot sequence these repetitive sequences, and some important genes interspersed regions, so that can be confirmed with the Sanger's only. So up till now, Sanger is also considered as a gold standard sequencing method. Sanger sequencing is still widely used for the small scale experiments. Means in a lab study, when we need a small, means 1000 or 2000 kb, when we need small scale experiments, then there will be no use of this NGS. Means NGS is used for the larger genome. So at that time, Sanger sequencing is very useful technique at the small scale experiments for finishing the regions that can be easily sequenced by the NGS, mainly highly repetitive sequence. Now DNA sequencing is widely used for the research purpose, like targeting the small genomic regions in the large number of samples. As I said in the previous, if we had a small sequences in a different sample, so many samples, then it can be done with the Sanger sequencing only. Sequencing of the variable regions means when there is a polynucleotide homodimer, means AAA, AT, AG, that long stretch of this single nucleotide or dimers that cannot be accurately sequenced with the help of NGS. In that case, Sanger sequencing is the important technique for sequencing these variable regions, validating the results from the NGS technique, like small gaps and important genes, and also verifying the plasmid sequence, insert, and mutations. Uh, Cloning technique also include the insert DNA of around the 1 kb, 2 kb, like that plasmid sequences that are sequenced with the help of Sanger sequencing. Also, the samples having some, we have at the lab level learned some manipulation like insert, mutations, site-directed mutagenesis that can be confirmed with the help of the Sanger sequencing only. Further, HLA typing means human leukocyte antigen, that matching between the two like parents and child siblings, that can be done with, and there is only a single nucleotide polymorphism or the group of the single nucleotide polymorphism checking that can be done with the help of the Sanger sequencing only. Genotyping of microsatellite markers, again these microsatellite markers are of around few hundred base pairs only, so that can be done with the this sequencing technology only. And identifying a single disease causing genetic variants means two individuals having the same gene but having a different condition, like disease condition, and one is the normal. So we need to find out what is the reason at the genetic level, so that particular SNP or the group of SNP that can be confirmed with the help of this Sanger sequencing only. The Sanger sequencing applications further at the advanced level can say that CRISPR gene editing confirmation for accurately accurately know the CRISPR gene editing further can be confirmed using the Sanger sequencing only. And as I tell, NGS validation, then Sanger sequencing using ion ampli-seq primers and libraries. Ion torrent is also one of the next generation sequencing technology that do the amplification of whole genome map. Here also some region like minor alleles, some specific genes that can be confirmed with the Sanger sequencing. At a low level, somatic variant detection at the tumor samples, one normal healthy samples and the new tumor samples. Here also some gene or some particular SNPs that are different between normal and tumor cancerous cells. So that can be comparison, and that detection can be confirmed with the help of Sanger sequencing. And genotyping of HIV-1 to detect the drug resistance. There is a genotyping of genetically diverse virus of this HIV virus strain can be detected. Genotyping of this HIV virus can be done with this sequencing technology. And Sanger sequencing solution is also used for this SARS-CoV-2 research in the lab. Now, these examples demonstrate how the Sanger sequencing has kept its value to researchers over a decades. and it is an enduring technology that deserves a place in a toolkit of every scientist. Indeed, we might still be using it another 40 years. Thank you.
Okay. So now it's too informative and valuable productive. Now I would like to invite Dr. Ajit Jadhav sir for the vote of thanks. Sir, please.
Okay. Thank you, Dr. Punjab, Michael ma'am, for sharing informative webinar on the sequential DNA for the information of the students and the scientists and the professor of the College of Agriculture and Parul University. We are very much thankful to Dr. Punjab. Thank you. Thank you.
I'm also thank you, sir. I'm very grateful for Parul University and to you for giving me these opportunities for giving this online webinar. Thank you so much, sir.
Thank you, ma'am.
Thank you, sir.
Okay. Webinar team, we can end this webinar.
Thank you, Dr. Sayed, sir.
Okay. Thank you, ma'am.